Good afternoon, everyone. I'm Dr. Manoj Malik, and I'm going to present my paper titled Host Polymer Matrix Dependent Significant Improvements in Electro-Optical Switching of Polymer Dispersed Liquid Crystal Films. So this is my presentation outline. First, I'll be talking about liquid crystals, the classification, brief about the properties and certain applications. Then what do we mean by polymer dispersed liquid crystals? That is coming out from the liquid crystal term itself. Then objectives of the present work, experimental measurements and results, and then conclusions. As we know, the matter exists in three elementary forms, that is solid, liquids, and gases. Liquid crystals is often known as the fourth state of matter that find itself in between the crystalline and the liquid phases. Now, similar to the crystals, the liquid crystal molecules have some kind of orientational order and positional order. They are anisotropic, and similar to liquids, these liquid crystal molecules have the freedom to flow. So they appear to be in a fluid phase, basically. Okay, and they also often, the XRD results of certain liquid crystal molecules also show that they often are found to have 0, 1, or 2-dimensional lattices. So generally, liquid crystal is another state of matter which is in between the crystals and liquids. Often also it is known as the floating crystals because they have certain degree of flow like liquids. Now, broadly, liquid crystals are classified as thermotropics and lyotropics. Here in my presentation, I will be talking about the thermotropic liquid crystals. Based on the temperature response to these liquid crystal molecules, they are classified as nematic, polystric, and smectic. Now, these gray cylinders, these are molecules. These are elongated molecules. The liquid crystal molecules are generally elongated. And so these molecules are more or less arranged in this order. And if we overall have to define a director, that is the direction of all these molecules pointing in one direction, so we can denote it by vector n. So this is the nematic director of these liquid crystals. In polystric, we see there are layers of these stackings of liquid crystals, and each layer has one particular orientation. And in smectics, they are more ordered. Okay, and then we have many other kinds like ferroelectric, anti-ferroelectric. Now, recently, the newly discovered liquid crystals are blue phase liquid crystals. And one of these liquid crystal phases, that is the discoitic or the columnar phases, was invented by Indian scientist S Chandrasekhar in 1977. So Indian scientists also have a contribution to this field. Now, what are the general properties of liquid crystal molecules that attract us and that make us to focus on their applications? First, they flow and form surface level similar to liquids. The most important property that I'm going to talk about is the second property. They exhibit anisotropic material properties just like several solids. For example, optical birefringence. Now, what do we mean by optical birefringence? That along the longer axis and along the shorter axis, the measurement of refractive indices is different. So this gives rise to the non-linearity in the optical measurements or the optical birefringent properties. Similarly, because of their anisotropy, they also show dielectric anisotropy. And because of all these properties, these liquid crystal molecules in the presence of electric field have this property to rotate or align themselves depending on the positive dielectric anisotropy or negative dielectric anisotropy. For example, in case the molecule is positive dielectric material, liquid crystal, it will align itself parallel to the electric field. And if it is negative anisotropic material, it will align itself perpendicular to the applied electric field. Now, what is the significance of all these properties? So here comes the most important part, that is, just by the applied electric field, tuning the applied electric field or having a control over the applied electric field, the liquid crystal molecule can be made to rotate inside a material, thereby controlling the refractive index of the or the velocity of the light passing through it, or in other words, optically modulating the light passing through these liquid crystals just by applying electric field. Okay. And based on these important properties, there are many applications of liquid crystals. Like we all know, all the liquid crystal displays, these LCD devices, these are nothing but containing these liquid crystal molecules. Then Since I spoke about in my previous slides, these are thermotropic molecules, so they respond to temperature. So based on those properties, we have liquid crystal thermometers or temperature sensors. Then switchable smart windows. These are a class of relatively new class of materials, smart windows, under which we have polymer dispersed liquid crystal. That will be the focus of my topic that I'll be talking later. Then optical imaging also, because of their selective, just like in this slide I showed, there is this layer of liquid crystals. In each layer, they are orienting in one direction, but helically they are arranged one above the other. So this gives rise to a pitch of the liquid crystal, and selective reflection and transmission of light can take place, giving rise to colors or optical imaging. So in this slide, I talk about the PDLC. So generally, liquid crystals are embedded in the polymer matrix by certain methods. So they form this composite film known as polymer dispersed liquid crystals. And these PDLCs find a range of applications in flexible display devices, smart windows, phase modulators, polarizers. Polarizers, definitely, there is some alignment process of these liquid crystals. And the switching effects can give rise to polarization of light, light switches, optical shutters, switching gratings, and temperature sensors. Now, as I already spoke, generally PDLCs. Now, the important thing is when polymer is involved, liquid crystal molecules are involved, and we are talking about the optical transparency or the light passing through these materials and optically modulating the light. So there has to be electrical applied field, and which is not possible without introducing an electrode to these materials. Now, the moment we introduce electrodes, the entire assembly becomes opaque. So what is done basically for this green part, suppose this is polymer and these are the liquid crystal molecules, the pale yellow droplets inside. If these are dispersed in this green polymer, now for the incident light from top, it has to pass through the entire material. And also, whenever we have to electrically control or we have to apply the electric field, even that provision should be there in the assembled the assembly. For that, we use ITO coated indium tin oxide coated glass plates as electrodes on both the sides of the composite material. So this way, this glass is not a normal glass; it is a transparent electrode. Okay, so this is ITO coated glass plates through which we apply the electric field, and then we observe the modulation of light after applying some electric field. In general condition, without the application of electric field, during film preparation, the liquid crystal molecules are oriented in random directions as shown in the A part of the figure. So when no electric field is applied, the light incident encounters certain scatterings and certain deviations from its original path, giving rise to a opaque appearance. It is not opaque, but it is translucent. Okay, it is translucent appearance with the application of electric field, because these liquid crystal molecules have the tendency to align themselves in the direction of electric field. The light then passes easily through these molecules without uh, encountering any kind of refractive index mismatch. I have, sir, two videos, the films that we have prepared in the laboratory. So over here, we can see this is a film, which is a liquid crystal PDLC composite film. Over here, the electrode pins are applied to this part of the glass plate, forming the lower electrode, and this black pin is clipped to the top part of the glass plate, forming the upper electrode. And in off state, we can see it is a kind of hazy appearance or opaque appearance. As we increase the voltage, see the picture behind this glass plate, there is a picture behind this glass plate which appears to be, which is now visible as the voltage is increased. Okay, so this is the light transmission changes that are taking place through these composite films. Okay, we can see the picture is pretty much very clear. Now, if we reduce the voltage, again the molecules, they return back to their initial state of random orientation. At that time, this picture, again, the film becomes opaque and the picture no longer can be seen through the device. Against light, over here, I have another video where we can see these films are kept in front of some numeric digits having a light intensity behind them. And if I increase the voltage from the left side in the digits, we will see slowly the figures behind the films are visible. Here, the film becomes clear. as we increase the voltage. And as we decrease the voltage, again the film becomes opaque or translucent, basically. So it is overall just a electrically controllable switching or modulation of optical light of the optical modulation of light passing through these devices. Now, what are the objectives of the present work? Very often altering the conventional PDLC films not to, because it is involving a lot of electrical applied field, light modulation, everything. So definitely there have been efforts to reduce the power consumption, the voltage applied, and to have the films performing better in ambient conditions. For example, temperature, over a wide range of temperature. So generally, the conventional PDLC films composing only of polymer and liquid crystals are generally uh, they are added by certain other foreign moieties to increase the performance of the film. For example, liquid crystalline polymers, copolymers, UV filters, surfactants, electrochromic molecules, and variety of other molecules. But what happens is such methods often lead to relatively complicated film preparation methods, free radical generation, some ionization in the film, affecting the overall applied field, the aging related issues, the switching times, the voltage applied to the film, the minimum voltage applied, and there are so many complications. So in this present work, we report a simple method to formulate the only we are changing the host polymer matrix composition. We are changing just the host polymer matrix combination. We are adding no dyes, no outside foreign moieties or molecules, no ions, no nanoparticles, nothing of that sort. We are just modifying the host polymer matrix with varying combinations of PMMA and PMMA-BA. So one is a polymer polymethyl methacrylate and one is a copolymer of PMMA-BA. Now, these materials were chosen on the basis of their physical properties that I'll talk later. And then the effects of these polymers on the morphologies, the transmittance, the threshold voltage, the hysteresis effects are discussed. What we found a significant improvement in the electro-optical behavior of these devices. So these are the materials used, a monomer unit of PMMA, which has a glass transition temperature of 95 degrees Celsius, and the refractive index of this polymer was 1.4985. The copolymer, the glass transition temperature 99 degrees Celsius, and the polymer refractive index 1.5185. Now, carefully, why we are choosing why we are mentioning the refractive index of polymer? Because this has to match with this value over here, 1.5216 of the liquid crystal molecule when it will be aligned. It will be aligned fully with the electric field so that no deviation or scattering of light takes place. And the liquid crystal molecule that we are using, it's a commercially available molecule manufactured by Merck, eMerck. The commercial name is E7. It is nothing but a eutectic mixture of several other components, which are given over here in the figure. For both these materials to form a composite film, we have to identify a common solvent where these can be dissolved. So we use the solvent-induced phase separation method where the common solvent was chloroform. And how do we manufacture that? We have this common solvent, the green color, for example, the polymer and the liquid crystal mixture. It is poured on the glass plate, and the thickness is controlled by the spacers on the edges of this assembly. These spacers give us the freedom to adjust or decide the thickness of the film. Currently, in this present study, we have used 60 micron PET spacers to get the PDLC composite films of 60 microns thickness. Now, one of our main aim was to view the
timing is about than 14 minutes. Please compile.
Okay, sir. So I'll quickly come to the conclusion. We can see the homogeneous distribution of these liquid crystal droplets in all the composites that we have prepared by varying the PMMA and PMMA copolymer weight ratio. To confirm, these are the optical polarizing microscopy images at different voltages, for example, 0 volt, then 20 volt, 40 volt, 60. We can see the topographical changes as the electric field is applied. So light passing through also has some changes. On the right side, we have the SCM. We just wanted to confirm whether the material, the liquid crystal, is also in the bulk of the material. So we cryogenically fractured these films and observed under the SCM, and we found the favorable results that the method used is can be utilized further to prepare such films. Now, here comes the important part. The significant improvements are, for example, this black line is the pure PMMA film and liquid crystal, and this is blue line is pure PMMA, the copolymer and liquid crystal. In between, we can see we can improve the properties from copolymer drastically by adding certain component of PMMA in that without much what you can say compromisation on the switching transmitters. Threshold voltage and switching voltages were measured, and we found them that in case of pure PMMA-BA, that is copolymer, the on the extreme right side edges of the graph, the values are too high, and we can control them by mixing other polymer PMMA. Hysteresis measurement is scan-up cycle, increasing the applied voltage and then decreasing and measuring the transmittance. We can see on the right side figure, this hysteresis gap is significantly reduced when we have the PMMA component added in the polymer PMMA with the liquid crystal E7. So the conclusions was a simple and convenient method was reported, wherein the host polymer matrix was successfully formulated at a fixed loading of liquid crystal, that is 30%, that is 70%. The film showed good distribution of phase separated LC droplets that was concluded from the SCM and POM morphology. Electro-optical switching showed good voltage transmittance behavior of the composite films. Threshold voltage switching voltage were improved significantly. Lower value of hysteresis achieved at higher temperature value, because the bottom graph, the x-axis is the temperature. Even at high temperature, the film is performing satisfactorily. So these were the conclusions. Thank you for hearing me patiently.
So thank you, and today good afternoon to everyone. And today my talk is about polymer brushes role in stabilizing colloidal suspensions. And the plan of the talk is the following. I will first of all talk about what is the meaning of colloidal stabilization, and then a brief cross-grain view of a polymer chain. Then I will talk about real chains, a brief about Fleury-Huggins theory, and then excluded volume interaction and solvent strength, polymer brush, theory of flat brushes, and then Darjougin approximation and results and conclusions. I will go through them very quickly so that it can be finished in time. So first of all, what is the meaning of colloidal stabilization? If nanoparticles are colloidal particles means particles having shape, size, let's say in the micrometer range. If they are dispersed in a solvent, what happens is that strong van der Waals attraction create agglomeration. That means the whole solution becomes unstable. And it can be seen in the figure that below the two figures here that basically the system has phase separated because the solvent and the solutes has been separated from each other. Now, in that case, we need stabilization. The stabilization means we need colloidal particles to be dispersed uniformly throughout the solvent. And how can we do that? The remedy is that we can use surfactants, or we may use polymer chains grafted on the nanoparticles or colloidal particles. And what happens is that when the steric repulsion occurs between the polymer chains, the nanoparticles or the colloidal particles stay far apart from them in order to reduce the overall free energy of the system. And it can be shown for a polymer system. Okay, now let me go through briefly about the cross-grain view of a polymer chain. And here I have an ideal chain very briefly. Ideal chain means there is no interaction between the chains. And in case of a polymer, we basically measure the end-to-end distance, and which is given by R square is equal to NB square. And then we have the equilibrium distribution of the end-to-end vector, because the polymers may have many different conformations, and from that we can calculate the equilibrium distribution of the end-to-end vectors. Am I audible, sir, or visible?
Yes, still audible.
Okay, thank you. So equilibrium distribution of the end-to-end vector, and this is the expression which is the Gaussian distribution. And from this distribution, we can basically calculate the elastic energy of a polymer chain. This energy is basically entropic in nature, because in ideal chain there is no interaction, and therefore the whole elastic energy comes from entropy. We can also define radius of gyration for a polymer chain. These things, all these things may not be important for the present talk, but knowledge about them creates a sense of intuition when we go through the rest of the talk. Now, what about a real chain? Real chain means we have intermolecular interaction and also interaction with the solvent. And based on this interaction, we can divide good solvent, poor solvent, and a theta solvent. Good solvent means we have a favorable interaction with the solvent and the monomer. And in that case, the chains basically swell in the solvent. And poor solvent means we have an unfavorable interaction between the monomers and the solvent. And in case that the chains collapse. And we also have the theta solvent. And it is a situation where the monomer interaction and the monomer solvent interaction, they basically, balance each other. And as a result, we have an ideal chain behavior. And based on these real chain properties, that means we have the interaction with the monomers and the interaction with the solvent. Flory and Huggins derived what is known as the Flory-Huggins theory. And in this theory, they calculated the mixing free energy. And here is the expression for the mixing free energy. And here, phi is basically the volume fraction of species A. I have written them as species A and B. They can be polymer, they can be, nanoparticles, or they can be, and B species B may be any kind of solvent, isotropic solvent or anisotropic solvent. Na is basically the number of solvent segments in the species A, and Nb are the number of segments in species B. And chi is a very important parameter, which is known as the Flory interaction parameter. And this parameter basically describes the interaction between the monomers of the polymers, or the monomers and the solvent, or the solvent-solvent interaction. So all kinds of interactions are included in the parameter chi. And for example, here we have a polymer solution. And for a polymer solution, at the end of the slide, we can see that the Flory-Huggins theory for the mixing free energy simplifies considerably for a polymer solution, because in that case, we have polymer and a solvent. Now, this Flory-Huggins free energy for a polymer solution can be further modified or further simplified if we assume that the volume fraction, that is the amount of polymers that is there in the solvent, is very low. And in that case, we can expand the Flory-Huggins interaction energy. And from that, we can extract two important parameters. One is called the excluded volume interaction, which involves the interaction parameter chi. And another one is W, which is called the three-body interaction parameter or ternary interaction parameter. And based on this excluded volume interaction B, we can also define good solvent, bad solvent, or theta solvent, just we defined in the situation earlier. And there is also an athermal solvent, in which case the chi is 0. Now let me come to polymer brushes. What is polymer brush? Polymer brush is basically if we tether polymer chains to a substrate, and if the surface density of the tethering chains is very high, in that case the chains basically try to overlap with each other. And when they try to overlap with each other, what happens is that the steric hindrance or the steric repulsion causes them to stretch away from the substrate. That means the chains basically stretch away from the substrate. And here is the figure of a polymer brush. It looks like a brush and brush-like feeling. So this is what is known as the polymer brushes. And it is very important, and these polymer brushes play an important role in the colloidal stabilization. So next is, what about the theory of a flat polymer brush? It is called a flat polymer brush because the surface is very flat, in which the polymer chains are tethered. There are basically two, in general, theories which has been propagated for the flat polymer brushes. One is called the Alexander Degin brush, in which case it is assumed that all the brushes, when they stretch away from the substrate, they uniformly stretch away and attain a height uniformly. Let's say the height is h, which is called the brush height. But this picture is overtly generalized, because the polymer brushes may not stretch uniformly. Some brushes may stay below the polymer height h. Therefore, the polymer ends may have distribution which is spread from the substrate to the brush thickness throughout the brush thickness. I mean, here is the figure that in which the polymer brushes, the ends of the polymer brushes are not perfectly uniformly stretched. Some of the brushes, the ends of some of the brushes are lowered and some of the brushes are stretched. And that's why it is called a non-uniform brush. Non-uniform brush in the sense that the chains are not uniformly stretched. And for which A.N. Semenov proposed a theory in 1985. And this theory talks about an elastic energy and an interaction energy. And here is the form of elastic energy in equation 1, where Gy is basically the chain end distribution that I talked about. That all the chains are not uniformly stretched, but they are non-uniformly spread throughout the chain, throughout the thickness. And EXY is basically the local elongation of the chains as they stretch away from the substrate. And next we have the interaction energy. Here is interaction energy in equation 2, and it is the general Florey-Huggins interaction energy that I talked about, that how the polymer chains interact with the solvent. And since we have introduced 2 new parameters, which is the chain end distribution and this elongation parameter EXY, so they satisfy certain constraints. And these are the constraints that I have written here, 3, 4, and 5. So these are the constraints that these new parameters has to be satisfied. Here is the total free energy of the system, equation 6, which is the elastic energy and the interaction energy. And if we minimize this total interaction energy with respect to the constraints that I talked about, we get equation 7. And equation 7 contains a very important parameter, which is called the chemical potential. Here you can see, here it is the chemical potential. So from this equation 7, we can calculate the chemical potential using the Florey-Huggins theory in the good solvent case. The good solvent case that I talked about, the good solvent case, I calculated the Florey-Huggins from Florey-Huggins theory, the chemical potential. This chemical potential then can be utilized to calculate what is called the volume fraction of the polymer segments. And equation 9 is basically the volume fraction of the polymer segments. If we calculate the volume fraction of the polymer segments, then ultimately we can calculate the total energy of a chain. That is how much energy is stored. That is the elastic energy plus the interaction energy. And here in equation 10, we have the total energy of the chain. Now, if we assume that for certain reason, the brush has been compressed due to force, due to a particle, here is a figure that where the brush has been compressed. So if the brush has been compressed to a length small l, which is less than the brush height h, then we can calculate again the total energy of the chains. Again, elastic energy plus the interaction energy. And here is the total energy of a chain for a compressed brush. So we have got a total energy for a chain of a uniformly, for a uncompressed brush and for a compressed brush. And here comes the Darjoguin approximation. And here is the figure. We have 2, let's say, colloidal particles or nanoparticles of radius r. They have the polymer chains grafted on them and they approach each other. So can we calculate the interaction energy between these 2 particles? The Darjoguin approximation says that yes, we can do it. And this interaction energy is basically the difference is somehow related to the difference between the flat brushes, the energies of the flat brushes that we calculated, that is the compressed brush and the uncompressed brush. And this is equation 12. This is equation 12, how the Darjoguin approximation can be used to calculate what is called the interaction energy between 2 nanoparticles or colloidal particles. And since we have already calculated these 2 free energies, so this is the free energy difference that I have calculated for a flat brush. And here is the result, that if we calculate the interaction energy, if we calculate this integration in equation 12, we get equation 13, where in equation 13, I have basically normalized some of the parameters, for example, the nanoparticle radius, the nanoparticle separation, and we have got an analytical result in equation 13. This is a very simplest model that I have used. And if we plot this free energy with respect to this L, which is L is now the particle separation in the normalized or non-dimensional parameter. So if we see this curve is very important. And the curve says that if the separation of the nanoparticles or the colloidal particles are getting reduced, then what happens? The interaction energy is increasing drastically. And if you see that these R are basically the nanoparticle radius in the non-dimensional regime. So these are nanoparticle radius, and you see that as the separations are decreasing, the interaction energy are increasing. So what it tells us is that as the nanoparticles or the colloidal particles are getting close together, the interaction energy is becoming very large. And therefore, thermodynamically, the system will become unstable and 2 nanoparticles will stay away from each other in order to reduce this overall interaction energy. And therefore, this polymer grafting plays an important role in order for the particles or the colloidal particles or the nanoparticles to stay away from each other in order to reduce their interaction energy and uniformly dispersed in the whole solution. So here is the conclusion from the curve, that if we decrease the separation, the interaction energy increases, and with increasing radius of the nanoparticles, the repulsion is also increasing. And basically what we can conclude from here is that the polymer grafting is a very unique way of dispersing nanoparticles or colloidal particles uniformly in a solution. Thank you.
Please start.
Okay. First of all, thanks to the Gyan Bharati Mahavidyalaya and Chemical Research Society for the collaboration and giving this opportunity for the all young researchers, and also for me. I will take it my pleasure to present my work. Here, I am Dr. Malatesh S. Pujar, Assistant Professor, working in Kelly Technological University from Belgium campus. And quickly, because of the short time here, I'm presenting a short about the work, about my one of the part of a work here. That is a copper oxide and polyaniline multi-walled carbon nanotube. Actually, we try to synthesize the composite of the nanomaterial using a Macaranga Indica leaf extract, which is used for the hydrogen gas sensor. I will give a brief highlights of that one. We have synthesized this using a leaf extract, so you can consider this as a green route, of course. And also, we went for the certain morphological studies and the compositional studies as well. In morphological studies, we have studied for the SEM, and there we observed 30 to 40 nanometer of a size of that composite, and also we confirm that with the XID also. And in EDX, we have tried to check whether the purity is up to the mark or no, and then we gone for the thermal variations. There we observed a 17% and a 25% of a variation with respect to the copper oxide, and also with our synthesizer composite. And after that, we have gone for the hydrogen gas sensing for the two different temperatures with variation of a hydrogen gas concentration also, so that it will be very easy and helpful for which concentration we can prefer it as an optimum level. So that we varied from the 110 parts per million to the 2 parts per million for the two different temperatures that we choose for the 200 degrees Celsius and 250 degrees Celsius. And then after that, using the same data, we have calculated with a known logistic function, that the equation I have mentioned here in a presentation, and also we have already reported that with our paper article. It is already published with the analytical chemistry and also with the biointerface journal. And the sensitivity we observed from 5% to 20% as we go on increase the hydrogen concentration. Here quickly with the graphical representation, I would like to highlight how we have sized. This is a green extract that we considered by the Indica leaves, which is very well known everywhere. It is available. And we have collected these leaves from our local area. And by that, we have collected the leaves and washed that with the distilled water, and then chopping that, and all the very well-known matter, that is the dried leaves, are cutted and washed with the distilled water, and the 50 ml of that, of a 1 millimolar of a copper nitrate solution, was mixed with the whatever the plant extract we have got. And then the pH is maintained up to the 10 by adding the NaOH solution. And then we have irradiated that with our micro oven for the next further process as a centrifugation and the annihilation process. Ultimately, we got the powder. And that we have tried to mix with the multi-walled carbon nanotubes, because here we wanted to prepare just like a coating structure, so that we can create more porous or some kind of gaps in that, which will absorb or which will lead for the oxidation and reduction reaction, so that it can sense the gas whatever we pass to our material as a target. So here we prepared a separate solution of a 320 milligram of aniline hydrochloride for the synthesis of the further nanocomposites. And this is our brief process, whatever we have gone through for the ultimately to get our carbon oxide and Pani multi-walled carbon nanotube composites. And this is after getting the nanocomposite powder. First, we have gone for the XID, so that here we can confirm whether the peaks are well matching with the old literature, and those has to be carried further when we prepare the ultimate carbon oxide, Pani, and multi-walled carbon quantum dots. Here we can observe the pink line there. I have mentioned or put a star mark there, which is indicating the peak of the Pani. And this one we have cross-verified, and the same data sheet is matched with the JCPDS file that I have mentioned here, 800076 and 010646. By this one, we can confirm that we have synthesized the composite of the copper oxide, Pani, and the carbon nanotubes. And this is the functional studies that we consider as Fourier transform infrared spectroscopy. Here we have gone for the wave number of a range from 4000 to the 500, which is well known, and the broader peaks at the higher wave number, which are corresponding to the multi-walled carbon quantum dots, nanotubes, which are the presenting OH functional groups, and we have to cross verify with our pani also. There, that for that the peak is associated with the 1150, and then copper oxide stretching vibrations which are present for the lower wave numbers that we observed at the 525 nanometer. These are the some fine images which we are revealing the purity and the structure which we have prepared. The particles we have got, that is of 100 nanometer of a first, I mean the A image, it is representing the copper oxide nanoparticles which are taking very fine particles, and then the second one, it is representing the pani is coated, that means that is mixed with that one, so that it is losing its structural property and it is showing like a stone like a structure. And in a C, we can observe the multi-walled carbon nanosheets are started to mix up with that one. So it is again, it is not a proper structure. It is showing it is mixed up with some kind of nanotubes. And then when we introduce that with the combining the whole nano composite, here it is showing in a image D. There we can see it is a octahedral and it is coated with the pani as well as the multi-walled carbon nanotubes. And the E and F, the images representing it is a spectra of course, the EDX. There in a E, we can observe it is a pure form of a copper oxide, which we have prepared initially. That is the EDX spectra. And there we observe copper and the oxide only. And then when we compile the everything and prepare the nano composite, that is the representation of image F. There we observe the oxygen and the carbon also, because the carbon is introduced in the form of a multi-walled carbon nanotubes. And this is the thermal stability. And why this it was necessary means when we go for the apart from the room temperature, because the whatever the applications we are going to consider for the gas sensing and all, usually those all are going to follow the kinematic equations of a gas. So gas is very easily, it will rise its temperature and easily it will react with the when you are going to rise the temperature. So it is mandatory to study the thermal property or a thermal stability of our material, so that we can whether it is adequate for the further applications or no. So we went for the TGS studies. There we observed a 7% of our weight loss up to 100 degrees Celsius, and of course that is related to our water molecules. But further, if you go on rise the temperature, there we observe the 17 degrees Celsius for the copper oxide and the 25 degrees Celsius, which is not a remarkable degradation, because we have introduced a plant also there, so that biomolecules are going to be a reason for the degradation. And ultimately we didn't observe much loss, so no weight gain in a inert nitrogen atmosphere. This will suggest us it is going to be intact with the oxidation process. Yeah, this is important and the highlight of our main work of our work. This is the gas sensing. As we already informed, we have studied this one for the two different temperature, that is one for the 200 degrees Celsius, one for the 250 degrees Celsius. Parallelly, we varied with the hydrogen concentration also. So as we go on increase the hydrogen concentration, that is from the 100 parts per million to the 2 parts per million, there we are observing an increase in the resistivity. That means if you go on increase the concentration of hydrogen, then it is the sensitivity whatever we are observing, that is also increasing. If you decrease the concentration, it is showing that there is a lower of the gas absorption and also as well as sensitivity. There is a small inset I am trying to show there in image A, that is representing the variation of a gas, that is a hydrogen gas, whatever we have considered. The concentration we are increasing, then with respect to that the sensitivity is also increasing. So if you decrease the concentration, then sensitivity is also decreasing. That is why, because the availability of a hydrogen molecules, if our copper oxide nanoparticles behaving as a cations, then if hydrogen is behaving as an anions, then there must be interaction. So to have that interaction, the giving or a supply of a hydrogen is important. So we are varying our supply of a hydrogen. With respect to that, the sensitivity is also varying. And in coming to the second one, this is a response time was approximately for the 290 seconds, while recovery time is of 500 seconds. Here I would like to highlight, because for presently the recovery time and response time is useful for even for the catalytic and the conductivity studies also. So here I would quickly I would like to highlight the response time. It is the time needed for the variation in a resistance with respect to 90% of our equilibrium value. when it is exposed to the targeted gas that we have considered as a hydrogen. And the recovery time, what it tells us, the recovery time will give us the time which is needed, particularly when it returns to 10%. At that time, the resistance should be maximum when it is again exposed to the air. Apart from the hydrogen gas, when it exposed to the air, then it has to give the maximum resistance. This is our recovery time, and that we observe the recovery time 500 seconds and the response time 290 seconds that we have observed. And gas sensing response, that is a range from 1 to 1.28, that is at the bottom insert we have shown. And one more formula which is by the literature we have considered for the calculation of the parameters. And here, these are the 4 important points, particularly which we have observed. That is the key process of these sensors is copper oxide will be considered as a p-type and the n-type of a multi-walled carbon nanotubes. So the hydrogen gas was detected by the copper oxide nanoparticles is very less we can consider as per the literature, but here we didn't observe any hydrogen sensing by the copper oxide. So we started to do that or mix that to prepare the nanocomposite, and then we perform the experiment. There we observed really good result, that is for the different temperature with respect to the different concentration of hydrogen. Here what we have to understand by this work is when we introduce like a hydrogen or NH3, H2S, they are going to bind with at the surface of the oxygen anions. And here hydrogen we have used, that is started to binding with our surface of our nanocomposite. Their electrons started to pump out into the surface. This allows the depletion region diameter to be minimized and band bending to the reduce and resulting to the increasing conductivity. This is a representation of the same data with respect to the sensitivity. This is a bar diagram which is very easy to understand, so that we have gone for the bar diagram. As we, along with the x-axis, we can see there the hydrogen gas is increasing. With respect to that, sensitivity is also increasing. That we have again represented for the 2 different temperatures, that is a 250 degrees Celsius and also for the 200 degrees Celsius. Here, if the hydrogen detection behavior is determined by the redox reaction of our H2 and the chemiabsorbed oxygen molecules. But the hypothetically or by the literature, we have tried to write the equation, the probably or a possible reaction, what happens when it is exposed to the air. That is the equation I have presented here. And then the amount of the H plus ions is inhibited with our composite, whatever we have filmed, that will be prepared. We have prepared as a film, but here I would like to mention we can also do the same experiment by preparing the pellets, or else by the thin films, or else from the thick films. We can do as per our convenient and the availability of our lab facilities. And so that the H ions can recombine with the free electrons and they can decay into the hydrogen molecules. When it is subjected to the hydrogen gas, then what happens? H2 will be absorbed by the copper plus 2 atom, and then the whole nanocomposite film will be decomposes to H plus ions. That means it will send some kind of electrons so that it can lead for the conduction. So we can observe the increase in the conductivity. As a result, the level of H2 goes on increases. So the H2 ions are increasing, so that electrical conductivity also increases. So if it is increasing, then resistance is going to decrease. The same thing we have observed in our earlier graph. And this is just a part of my work. I tried to finish it or highlight based on the sensitivity, whatever we have observed. The same work we can do with the other gases also. And now this part is, I'm not going to reveal completely, because we are trying this one with the other metal oxides, with the rare earth metal ions, which is really not reported yet. So we are working on it, and we reported for the patent also. So I'm not up to reveal the complete work. And up to here, I would like to conclude and acknowledge some of the funding agencies. That is a CSIR UGC, which is given for, as a SRF and JRF fellow to one of my student, a research student, and the UGC and NSRIB, and the instrumentation facility at the USIC KUD, and also at the KLE Technological University. And one more, the main important funding we got from the RUSA. That is, we have considered for the instrumentation. Here I am also given my mail ID. The students and the colleagues or what they can ask the questions. I'm ready to take, and for any further collaboration also, it is really welcome. That is my malteshpujar.mss@kletech.ac.in. Thanks for my time.
So very good afternoon to all of you. My name is S Tamgadge. I am presently working as an assistant professor in the Department of Physics, Mahatma Phule Mahavidyalaya, Varud, District Amravati in the Maharashtra state. Today, my topic of my presentation is synthesis and characterization of L-alanine passivated ZNS nanoparticles, zinc sulfide nanoparticles. So let us discuss first of all introduction. So what is nanotechnology? We all know nanotechnology deals with the materials with the particle size having at least one dimension into the nanoscale. And why we are working into the nanoscale? Because it has various applications due to the high surface to volume ratio and also the quantum size effect. We all know that. And zinc sulfide is nothing but a metal sulfide. So why we have chosen this particular material is because it is having advantages in the various NLO devices. And zinc sulfide is having a wide bandgap energy of a 3.72 electron volt. And again, we are reporting this paper with the passivating agent, which we use as amino acid. So why we have chosen this amino acid, and in particular that L-alanine, because of the easy availability, its low cost, its biodegradability, non-toxicity, and environmental friendly properties. So this is the abstract of my presentation. We report the synthesis and characterization of ZNS nanoparticles passivated using L-alanine amino acid by simple co-precipitation method at room temperature. The samples in the powder form were characterized by X-ray diffractometry, EDS, UV visible spectroscopy, TAME, and also FTIR. XRD and EDS confirm the formation of ZNS nanoparticles with the desired phase and purity, and ZNS nanoparticles with average particle size of 4 nanometer were observed for
Tamgadge, we are still seeing the abstract slide.
Okay, yes, sir. Yeah. Is it visible, sir? Next slide.
Yes.
Yeah. So in this study, what we have done, we have synthesized pure and L-alanine cap ZNS nanoparticle by simple co-precipitation method. And these particles have been characterized by XRD, EDS, and HR-TAME. We have also studied the optical properties of these nanoparticles by using UV visible and FTIR spectroscopy, and the results are presented over here. So this is the experimental section. This is very simple. It can be seen by the various papers, and we have adopted the same methodology. We have used zinc chloride, sodium sulfide flakes, ethanol, and with all AR grades. They were procured from SD Fine Chemicals, Mumbai, and L-alanine was purchased from the Sigma Aldrich, USA. For the synthesis purpose, this process involves dissolving a salt precursor, usually chloride. So we have dissolved zinc chloride into the water, double distilled water. The corresponding metal sulfide usually form and precipitated in water by adding the sodium sulfide to the solution. The resulting chloride salts, that is NaCl, are then washed away, and the hydroxide is calcined after the filtration and washing to obtain the final oxide powder. So these are the samples which were prepared by us. So we have prepared 4 samples actually. So first sample is ZS pure, means without capping agent. This ZS1 sample was prepared by using the capping agent with the quantity of 1 ml of 1 molar concentration. This ZS2 sample was prepared by using the L-alanine capping agent with a quantity of 2 ml. And this ZS3 sample was prepared by using 3 ml by putting 3 ml L-alanine of 1 molar concentration. So these are the results. So let us first discuss the XRD. So these are the X-ray spectra for 2 samples actually. We have recorded it for 2 samples, one for ZS pure and another for ZS3. Remember that ZS pure sample is a sample which was synthesized without using the capping agent, and this ZS3 is a sample which was prepared by using a capping agent of highest quantity. So from this XRD, we can see that the presence of zinc blend phase of crystalline ZNS with all cell parameters A, B, C, which are mentioned here. And there is no peak due to the impurity, which can be seen from the XRD. And we have calculated the average particle size, which was found to be 4 nanometer for the sample ZS3 and 7 nanometer for the sample ZS pure. So this XRD pattern confirms that by using L-alanine as a capping agent, the particle size is greatly reduced. This is the EDS study. So from this, we can see the purity of the sample. Only the peaks related to the zinc and the sulfide, these are shown here, and no impurity peaks were recorded. These are the images of HR-TAME. So from this also, we can see that near spherical particles have been synthesized. This is the UV visible spectra. So from this, the maximum absorption peaks can be seen over here. So for the ZS pure sample, the peak was recorded at about 320 nanometer, and for ZS3, the peak was shifted towards 295 nanometers. So the ultraviolet shift is there. So we can conclude that the particle size is decreasing by using the L-alanine. This is the FTIR spectra. So all the functional groups related to the L-alanine were observed and were confirmed. So in conclusion, what we can say that L-alanine capped and uncapped ZNS nanoparticles have been successfully synthesized by the at room temperature by co-precipitation method. Strong blue shift in the UV visible absorption spectra is seen for the sample capped with L-alanine, confirming the role of capping agent. UV visible absorption spectra show maximum absorption peak from 320 nanometer to 295 nanometer. XRD, EDS, HRTM data show the formation of ZNS pure phase with no impurity content. Peak broadening can also be seen, and it confirms the formation of narrow crystalline size. The average particle size from the XRD is found to be 5 nanometer. The FTIR spectrum confirms the vibration of ZNS and L-alanine components. Obtained nanoparticles may be potentially used in various optoelectronic devices. So, thank you.
Yes, thank you.
Yeah. So, good afternoon, everyone. So, myself, Dr. Pankaj Arora from BITS Pilani. In today's talk, we are going to discuss about 2D nanomaterials in plasmonic sensors for medical diagnosis. So, before going to how we are utilizing the 2D nanomaterials for the sensing purposes, what we are going to show first how to affect the surface plasmon. So if we go in the literature, the first configuration which we came across, the Crashman configuration. So the Crashman configuration, what does happen? We use a generally glass prism, coated with a thin metal layer, and usually the conventional metal being used is gold, silver, and upon that we put an analyte. So the basic mechanism of exciting the surface plasmon, I'll get to that what is surface plasmon. So the basic mechanism is the incident light, polarized light, and everyone is familiar with the total internal reflection phenomena. So at the angle greater than critical angle, everything should get reflected back. Yes. So if I'm seeing the reflectivity curve, what should happen? Everything should get reflected back. Yes. But since there are free electrons in the metal, it's gold, silver, at one particular resonance condition, the whole energy of this light will transfer to the electron and they start to oscillate. The oscillation of this electron is called the surface plasmon. I'll get back to that. What is that condition? When does it happen? So the condition is when the wave vector of the incident light gets smashed to the wave vector of the surface plasmon. That's what I wrote it down over here. So the left-hand side, the wave vector of the surface plasmon, and the right-hand side is the wave vector of the incident light passing through the prism at a particular angle of incidence. So as I told you, one particular angle, the light will transfer its energy to the electron. That means at that particular angle, I should not get anything in the detector. Yes, everything should be transferred to the electron. So in the reflected light, I should not get anything. And as a result of which, for particular that angle, I should not get anything. In the reflectivity curve, it should be close to the zero. So that is how we are going to utilize this technique for a sensor for medical diagnosis. So prism index is fixed, wave vector incident light is fixed. At one particular angle, metal which I'm going to use, the dielectric constant is fixed. The analyte dielectric constant which we are interested in, depending upon how much is the angle, at which angle I'm getting reflectivity minima 0, I'll get to know what is the analyte index. So if we see the literature, there are two configurations. One is called the angle interrogation, other is called the wavelength interrogation. As the name suggests, angle interrogation means I'll be capturing the reflected light with respect to the incident angle. On the x-axis, I have the incident angle. So what is going to happen? As soon as I change the refractive index of the analyte, glass is fixed, metal is fixed. As I change the index of the refractive index, 1.33, I'm taking the example from starting from water, 1.34, 1.35. If I change the analyte index to match the condition, theta has to be changed. The angle of incidence has to be changed. And you can clearly see how much is the change. And if I'm defining a sensor, there should be some sensing parameters. The first parameter we come across for a sensor is sensitivity. So if I'm talking about the angle interrogation, the sensitivity is how much is the change in the angle, delta theta, with respect to how much is the change in the index, delta Na. So delta theta divided by delta Na would be sensitivity proposed under the angle interrogation. Moving to the wavelength interrogation, name suggests, now my theta is going to be fixed. only the wavelength is going to be changed. So wavelength is going to change. What does that mean? That means instead of using a single wavelength source, I'm going to use a broadband source. Yes. So what is going to happen? Now my theta is fixed. Now this wavelength is going to changing with respect to the change in the analyte refractive index. The same thing, Na is being changed. Now on the x-axis, if it is wavelength interrogation relevant to spectroscopy, I'll be having the x-axis wavelength. How much is the change in the wavelength? Delta lambda. With respect to how much is the change in the refractive index? Delta Na. That's how I'm going to propose the sensitivity for my angle interrogation. Now, so far, the literature we came across, people are mostly using gold and silver. Aluminum nowadays coming into picture. We choose in this work the aluminum to make a sensor cost effective. Moreover, the aluminum refractive index in the near IR region gives us a very narrow line width. I'll get back to that. Why do I need the narrow line width? Before that, what we did, I'm just going with the conventional configuration we discussed in the previous slide. I'm having over here glass prism. I just flipped the geometry so don't get confused. So glass prism, aluminum metal, analyte. Light is coming from a particular wavelength and the reflected light is being captured. So the wavelength is only single wavelength. That means I'm interested in the angle interrogation. So what we did, reflectivity with respect to different incident angle to optimize how should the thickness, how much should be the thickness of the aluminum film. We try to change the thickness 20, 30, 40, 50, what we are interested in. The whole idea was the whole light should get transferred to the electron. That means in the reflectivity I should get almost zero. Almost zero. That is one of the decisive parameter, the criteria that what should be the optimized thickness. The thickness which will give me the minimum reflectivity, and the other parameter I'm going to define is called the full width at half maxima, the line width. The narrower is the curve, the more is the resolution offered by the sensor. So that term is called full width at half maxima. So what we did, we plotted to calculate trade-offs that R minima should be zero, very, very close, and the FWHM should be as small as possible to get a optimal point. The optimal point we found at 30 nanometer. So 30 nanometer in my next onward slides will be using the aluminum thickness fixed 30 nanometer. So now I'm going one by one. So the first objective which I'm going to propose over here, the graphene as a 2D nanomaterial in plasmonic cell for leptospirosis dissection. So prism is same, incident like 15 nanometer, aluminum we just fixed 30 nanometer. Now what we are proposing over here, silicon graphene stack in this proposed configuration. The graphene, we know that 2D nanomaterial higher absorption efficiency, so it can help us to bind the biomolecule or any particular molecule to the sensing surface. Silicon, a high dielectric constant material, will help me to increase the sensitivity. So before going to the work, I'm just putting up a table to compare our proposed results with the previously reported results. They are using the prism-based configuration. The sensitivity they are getting this 2-digit number, 33, 95. Of course, they are getting 137. We go to the stack property 200. The other parameter which I'm comparing over here, figure of merit. Figure of merit is the new parameter which I just defined, which is basically sensitivity divided by the line width, the FWHM. FWHM I want very, very small. Sensitivity I want very, very high. So sensitivity divided by line width, FWHM should be very, very high. Figure of merit should be very, very high. And we got 95. So coming back to the result. So when we try to find out the reflectivity with respect to the incident angle for the proposed configuration where I having silicon graphene sets, we found these kind of curve. So in this particular curve, we got 3 dips. Yes, so we are interested in the surface plasmon. Then we try to figure it out, what are those different dips corresponds to. We try to simulate those with the electric field Composite multi-field software, and what we found, the SP mode should always propagate along the metal dielectric interface. This mode is SP mode. Other modes which are coming into picture, the dips, that was basically the waveguide mode. It's basically guided. So this mode very interested in. Now the question is, as I showed in the previous table, why you're getting enhanced sensitivity, enhanced figure of merit with regard to the conventional configuration? We try to, whatever the field put it was, we try to calculate the field enhancement with regard to the conventional configuration and the proposed device, and we could clearly see the enhancement with respect to the field enhancement at the interface. More is the field enhancement, more is the interaction of the light with the analyte, and more would be the sensitivity. Further to figure it out, we tried to zoom in that field plot, and we could clearly see the silicon graphene, white one is the graphene, red one is the silicon graphene stacks. You can clearly see the field got really enhanced with respect to the color bar. You can clearly see that was the main reason, the physics behind increasing the sensitivity just by adding silicon graphene stacks helped me to enhance the field, which resulting in a more interaction with the analyte means more sensitivity. Then we try to show over here with different detection concentration. Every concentration has a different refractive index. So there is a change in the refractive index, there is a change in the shift, and that's how we calculate the sensitivity and figure of merit. Now moving to my next objective. Just to confirm, I am audible and my screen is visible. Anyone? Am I visible and my screen is?
Yes.
Yeah.
Yes, it is visible.
So second objective, now we move to the next 2D nanomaterial, which is basically MOS2, molybdenum disulfide. So now what we are proposing, in the previous case, I was having silicon graphene stacks. Now I'm proposing MOS2 2D nanomaterial. Before going to the work, why we use MOS2? MOS2 is also 2D nanomaterial TMDC compounds. The MOS2 has more absorption efficiency as compared to graphene. It is having 2.5, MOS2 having 5%. More is the absorption efficiency on my sensing surface, more biomolecule will attach and more would be the sensitivity. So we tried to compare the same configuration with the previous reported results. So it is not only helping me to increase the sensitivity, but also helping me to improve the figure of merit as compared to my previous work. Graphene, I was getting figure of merit only 95. Over here, I'm getting the more figure of merit. So coming back to the work. So over here, we are proposing 4 different configurations. So I label them ASMG, aluminum silicon MOS2 graphene, aluminum MOS2 graphene, aluminum silicon MOS2, aluminum MOS2. So thickness is over here, they jotted down. So we try to calculate the sensitivity and figure of merit for all those configurations. So what we found, wherever I had the silicon, as I told you, silicon is a dielectric constant material. It helps me to increase the sensitivity. So the sensitivity, ASMG silicon is there, ASM silicon is there, G stands for graphene, M stands for MOS2. Wherever I didn't and where I introduced only the MOS2, over here you can see only MOS2. MOS2 helped me to reduce the line width of my curve. You can clearly see over here, the line width got reduced. Why? Because now in near infrared region, every material refractive index, we know that 1.33, 1.34 for water. But these 2D nanomaterials, they not only have the real part of the refractive index, but also have the imaginary part. And for MOS2, that imaginary part is very, very small. Smaller is the imaginary part of the refractive index, lesser is the absorption, which results in the narrower line width. So that's what we figure it out. Then to compare all the 4 configurations in terms of sensitivity, FWH and FOM, but we plotted and we found out that blue curve with respect to ASMG, if I'm going with the higher sensitivity point of view, the ASMG is the best option. If I'm going with the figure of merit point of view, because the MOSC giving the narrow line width, AM is the best option. So with these 2 configurations, ASMG and AM, we try to measure the different concentration of DNA. With different concentrations, you could clearly see there's a shift in the surface plasmon angle. So that is the second task. And I'm left with around 4 to 5 minutes. So I'm finishing my third objective. So the third objective, we are done with the story so far. We went with the graphene, 2.5% absorption efficiency, figure of merit 90. We went to MOS2, 5% absorption efficiency, figure of merit 3-digit number. The third one is the magazine. So magazine is a new popular 2D nanomaterial. In this particular work, we are not only using magazine, we are using the heterostructure made with the magazine. Why? Because magazine gets oxidized very, very fast. So to make it, to protect it from the oxidation, we took different 2D nanomaterial, make the heterostructure with the magazine, and that is the work we are proposing over here. So we again compare with the previous reported result. Now over here you can see, Pankaj, the sensitivity over here is higher as compared to your reported result. I completely agree. But see the figure of merit. The line width is very, very high. As a result of it, the figure of merit is very, very less. The idea was not only increase the sensitivity, but increase the FOM at the same time. So what we did again, but the only difference is over here I'm adding magazine black phosphorus, magazine graphene, magazine MOS2, magazine FZ. In the similar way, we try to calculate the sensitivity and the figure of merit. Now the extra step we did over here is see the effect of, because when we deposit these 2D nanomaterials, the flakes, it can be 1 layer, monolayer, 2 layer, 3 layer, 4, and so on. So we wanted to see the effect of the number of layers. So what we found when we put the more number of layers, in case of BP and graphene, as I'm increasing number of layers, the reflectivity minima is increasing. Moreover, the line width is also increasing. Whereas MOS2 and fluorinated graphene, even if you change the number of layers by experimentally, if you found deposit instead of 1, you deposit 4 or 5, the R minima is constant, line width is constant. To further quantify whether really it is constant, so we try to plot figure of merit with respect to R minima, with respect to number of layers. So you can see R minima is increasing, the blue curve, FOM is decreasing, R minima increasing, FOM is decreasing. But in the case of MOS and FG, it is almost constant. The beauty is in using that FG and MOS2, since, as I told you, these materials have very, very less imaginary part of the refractive index, it helps me to provide the narrow lane. So while fabricating, I don't need the stringent control, like I need to go with only 1 layer or 2 layer. Whatever the number of layers, the sensing parameter is almost constant. So with this, I would like to conclude my talk. So the first one, we propose silicon graphene stack-based proposed device, which helped me to increase the sensitivity because silicon is dielectric constant material. The second one, MOS2-based device, higher absorption efficiency, lesser is the imaginary part of the refractive index, helped me to narrow down the line width and increase the FOM. And the last one, magazine retrostructure with fluorinated graphene, helped me that I don't need any stringent control of the number of layers deposit of the 2D nanomaterial. With this, I'm done. Thank you so much.